So it's my pleasure to introduce the next speaker, who is Dr. William Lees, who happens to be our fearless leader of the germline working group. So it's only fitting that he gets 20 minutes. William's also become a leader in this space and has led pretty much most of the development for OgreDB and a lot of the germline database development work that has come out of the AIRR. He's going to tell you about all the things he's learned in the last 9 years. And I expect he'll tell you lots of good things and then maybe some scary things too. All right, William.
Thank you, Corey, and thanks very much to the organisers for giving me the time to talk to you today. So I'm going to talk about the importance of good germline sets and also some of the techniques that we're using to improve them at the moment, and highlight some resources that you might find useful in your work, and also give you a few tips on how I think you should be evaluating a germline set if you're thinking of using it. So first of all, why bother? Well, we've heard quite a bit about this already, but the VD and J genes that combine to form a receptor gene have a great deal of allelic variation. And germline sets are effectively lists of all those allelic variants, so they capture the variation. And the variation is being linked to the response to disease and other immune stimuli. So it's important if you want to understand immune correlates to those stimuli. So what are the issues in today's germline sets? Well, first of all, there are quite a few sequences missing from even the best germline sets today. I mean, human IG, which we've studied perhaps more than anything else, we still find multiple what we call novel alleles, previously undocumented alleles, every year. And this affects annotation. I mean, even at the most base level, if you annotate a repertoire with a germline set that's missing key alleles in that repertoire, you get a misleading idea of the mutation levels as shown in the red bars here. And in TR, we know that all 6 of the CDRs are important in binding to the presented peptide in the MHC. If we overlook the polymorphism in CDR1 and CDR2, which can be a particular issue if we're working from a fairly short sequence around the CDR3 and imputing the rest of the sequence from that, then obviously we're overlooking important polymorphism. Another problem in today's germline sets is they have sequences in there that probably shouldn't be there. And what I regard as a key study that came out in 2008 identified nearly 100 sequences in the IG germline set that had questionable evidence and should probably be removed. And I mean, very questionable evidence. I'll go into that in a little bit more detail if you're interested. And again, having those extraneous sequences there can cause issues in annotation. All sequences to be misannotated to alleles that don't really exist, and that messes up clonal inference and so on. So to address these problems, we need high throughput next generation techniques that we can use to fill in the gaps, find the missing sequences, and the data that we generate from those high throughput techniques will let us raise the evidence bar and get rid of sequences that shouldn't be there. So let's look at a couple of techniques that we're using these days to fill in those gaps. And the first one I'll talk about is inference from AIRR repertoires, so finding germline sequences from AIRR repertoires. There are several tools available that can do this, and they use a variety of statistical techniques, but the key sort of underlying principle that they're working off is that, on the whole, particularly in unmutated repertoires, the unmutated sequence here will show up at a higher rate in the repertoire than mutated sequences from it. The AIRR community has supported this germline inference process for a number of years through establishment of the Inferred Allele Review Committee, AIRR. Matt Solin is the chairman of AIRR at the moment, and his photograph is on my slide because he's here today and in fact has a poster on AIRR. And I've put a few other photographs in here of people who've been involved in this work and are here in conference. That's what it means. There's a photo there. AIRR has inferred over 40 or over 50 alleles, something like that, since establishment. And those have augmented our germline set and made it into others as well. These two plots come from a tool called OGDB stats, which AIRR uses to evaluate inferences using the process that's set out in this paper. And that tool's available for use for anyone else that's interested. Another tool that we use is VDJBase. VDJBase was the brainchild of Gor Yari and is developed in his lab. And on the repertoire side, it has over 1,600 repertoires. And actually, that's a number that we expect to increase over the next year or two, thanks to involvement in the AIRR Knowledge Commons project, which is a project aimed at bringing various immune databases together. And there's a poster on that at some point during the conference, I think. So these repertoires, they're all annotated with a consistent pipeline, which I've put a brief graphic on there. The aim of this pipeline is to identify the personalized genotype of each individual, so the specific alleles of each gene and the specific genes that are in that person. And then to look for haplotyping, if possible, and identification of novel alleles. The pipeline's published. It's called AIRR these days. This is a new pipeline that we're working on. It's developed by Ayelet Perez and Greg Klein, who are here. And there are some demos and posters on that during the conference. And again, that's published. It's a great pipeline. It's something that you could consider using in your own work, particularly if you're interested in this area. It's a great pipeline anyway. So the second technique I want to talk about is genomic sequencing. And on the left here is a plot of human IGH locus, VD and J. And as you can see, there are a tonload of genes and pseudogenes within about a megabase of sequence, as well as these various pieces of structural variation. We probably haven't discovered all the structural variants that occur between people yet. I'm sure there's more out there. The V genes are in here, the green genes are extremely similar to each other in sequence, and also to many of the pseudogenes in red. And if you think about traditional short read whole genome sequencing, V gene's about 300 base pairs long, something like that. A short read is perhaps 60 or 120 base pairs long, so doesn't even cover the entire V gene. And so you can see the issue of placing those reads accurately within this assembly, given the similarity of all the genes that are here. And an approach taken in Corey Watson's lab by Oscar Rodriguez and others is to use long-read sequencing, in this case PacBio sequences. So here the sequences in blue are significantly longer than the V genes in red. Probably this isn't to scale, they're actually quite a lot longer. And that enables you to capture not only the variation, the SNVs within the V sequence itself, but also in the surrounding context. And those SNVs in turn let you haplotype the reads, so you can allocate them to the two chromosomes and eventually produce haplotyped assemblies, map those assemblies against a reference, which includes both the human reference assembly, but also a number of these structural variants that aren't included in the human reference assembly. And then you can read off the genes at particular locations within the assembly. So within VDJ-base, we have genomic data as well. We have at the moment about 150 human IGH and 50 IGL. We capture not just the core coding region, but also the regulatory regions and the leaders of the V gene. So there's lots of information there. It's all available to download or to look at online, various tools we've got. And these assemblies come from sequencing conducted at Corey's lab and other high-quality sources. We're very, very interested in collecting sequencing from many sources. To give you an idea of what might happen to this, over the next year or so, I'm hoping that we'll extend VDJ-base to cover all 7 loci and have a much higher number of assemblies that we have today. They're deliberately selected from a diverse background, by the way. IARC is formulating criteria for reviewing and accepting novel alleles from this material. Corey, just the other day, submitted 34 novel IGK alleles to IARC for ratification, and I think that gives just a little indication of what we might see over the next period. So to change tack slightly, just talk a little bit about what defines a gene in the first place. And I think if we want to say that 2 sequences are alleles of a particular gene, we have to be able to localize that gene in an assembly. So we have to say, here are its coordinates, and we have to be able to show that those 2 alleles map to those coordinates. And many alleles in today's germline sets don't have sufficient evidence to be localized that way. That obviously applies to any that have been inferred from a repertoire because there's no localization for them, but also to many that have been inferred by other means and confirmed genomically. You may well not have enough sequence to localize them. You may just have the sequence of the gene itself. So the fallback within human loci has been to use sequence identity. And in many cases, although they're similar, the different V genes are sufficiently different that you can be reasonably confident of mapping an allele to a particular gene on its sequence. And there are some exceptions to that, but they're exceptions that are known, so we can deal with that. But what do you do in other animals? And to give an idea of the sort of problems you can run into, this is a, whoops, sorry, wrong slide, wrong button. This is a, well, on the top right-hand side, which I can't point to too well, these are haplotypes of rhesus macaque IGH, and they're roughly aligned. And the right-hand side of that picture, the sort of right-hand 2/3, is where the V genes are. And you can see there very, very substantial structural variation between some of these haplotypes, much more than we've seen in humans at any rate to date. And that's giving those of us that are working in this area real issues in terms of coming up with a definitive reference assembly where every gene can have a coordinate. It's a lot of duplication as well. So there we have identified a lot of allelic sequences, but we can't confidently map them to genes. And the analog we're using is, for the time being, until we can narrow this down a little bit, is to cluster on the basis of sequence identity. So that builds what we call allele sequence clusters. And to do this, we're using a tool and a publication that developed by Eilert Perez and others called Piglet. And this shows what Piglet does if you ask it to cluster IGH allelic sequences. And you can see there's pretty much a one-to-one mapping between the known genes in human IGH and the clusters that Piglet comes up with. There are one or two areas where we don't get a one-to-one mapping, and those are the areas where we'd expect things to be problematic. And given what I've mentioned before, that not all these alleles are correctly mapped anyway in today's world, this is something that has proved to be tractable in terms of repertoire analysis. With a little bit of care, understanding that we may not always have that one-to-one mapping, we can use the tools that I showed you in VDJ-base to do genotyping, haplotyping, and novel allele inference. And based on this, in the germline working group, we developed this approach to what we call a community approach to developing reference sets, where we identify with a unique label every identified allele. We map into clusters as best we can using sequence similarity where we have to, and then gradually evolve the germline set over time using these unique identifiers for each sequence as a way of preserving transparency. So a little bit on what makes a good germline set. Well, I hope you've been persuaded by now that good germline sets capture the diversity of a species. And obviously they need to rely on high quality evidence, and preferably multiple sources of evidence. And one thing that I think is important in particular is to combine repertoire sequencing with genomic sequencing. And that gives you two different ways of looking at the same picture and picking up anything that is systematic error or missing from one approach or the other. Good germline sets are also strongly versioned because they are going to change over time. And they provide traceability to sources because we're going to increase raise the evidence bar over time as more evidence becomes available. And we want to be able to go back through the sets and eliminate any that don't meet our new criteria. So what should you look for when you're evaluating a germline set? Well, you may well be constrained, particularly if you're not working with human repertoires. You may well find that there's not very much in the way of supporting evidence for the germline sets that you can find. In that case, I hope you've seen some of the issues that you might run into, and also some of the solutions in terms of germline inference. One thing to pay particular attention to is this business of strains and breeds. And if you remember from Corey's talk a little while ago, is a cow really a cow, and all the question marks on the slide there. If you have a germline set that's been developed from just one subject, from one particular breed or strain, and you're looking at another one, you may find quite a lot of question marks there. The important attributes are very much the ones on the left-hand side here. But the one I would caution on is this large number of sequences. You look at two germline sets and you find one of them has more sequences for your species than the other. Does that necessarily make it better? Well, it depends how good those sequences are. And having the wrong sequences in a germline set can be worse than not having them at all. I hope you'll consider, if you are working in human, the AIRR germline sets, which we announced for human IG this year. We're also working on sets following the same principles for human TR, which should follow shortly. So we've done everything we can in producing these sets to follow the principles which I've outlined to date. And in particular, to use multiple sources of evidence to exclude sequences that are questionable. And we've also done various things to make these sets particularly suitable for repertoire sequencing work. There's a poster in the working group poster session, which was first authored by Eve Richardson, that goes into more detail about what those steps in the fine-tuning are. And I hope you'll drop by and have a look at that poster. There's also a great document which she's written recently to explain how to use the sets with various annotation tools. I think it'll be very useful. So just to underline one or two of the points I've covered again, which I think are useful in general AIRR annotation as well as in germline development. Genotyping and haplotyping, which are techniques which will identify the particular alleles in your subject and, where possible, assign them to specific chromosomes, as shown over here with the expression levels of each allele on each side. These are great tools that can help you get more information about your subjects and how the genomics vary between the subjects in your study. They're also very good ways of coping with noise and unexpected novel alleles in your repertoire. There are many tools. I've just listed the ones I'm familiar with here, which are also the ones that AIRR uses. So you can even get value from these tools without going out and doing too much work. You can pick up a pipeline that supports them. Novel influence, particularly important when you're working with a germline set that you have less confidence in, or if you're working with a population that you think is understudied, for example, a human population. And I just wanted to mention very quickly to those of you that develop tools, the IMGT alignment, so this is a codon-based alignment of the V region, which places each of the CDRs at the same coordinates and typically ends up with the second cysteine position 104. We have discovered exceptions to this. The field has discovered exceptions that don't fit in this alignment unless you include insertions. Now, there's nothing wrong with having insertions, but there are tools out there that assume that an IMGT gap sequence will always have the CDR1 at a certain location, CDR2 at a certain location, and a second cysteine at location 104. You download the IMGT gapped sequences for macaque, rhesus macaque, you'll find that isn't the case because they've decided to put insertions in them. And there are cases where insertions are necessary. So just a word of warning to tool developers, this may get worse. And it's a good idea, I think, to make sure that you design your tools so that they can work with insertions in sequences, even gap sequences. So that's what I have for you, and I want to leave a lot of time for questions because there have been a lot of questions today. But just briefly, thanks to all of the people that I've worked with and have produced the work that has gone into the various slides I've covered. And in particular to Corey Watson and Gor Yari, who have both given me enormous opportunities to work within their labs and to help them put together some of the tools that I've mentioned, like VDJ-Base and OgaDB. And also to others in the AIRR community. I haven't by any means listed all the people that have been involved in the AIRR community and the GLDB working group, just some of them. But just to say, it's a great opportunity, particularly for early researchers to get involved and really get into something and get on a good publication if you have the chance to work in a working group. So thank you, everybody.
So William's endeavoring to get us back on schedule. Big task. We're way off schedule. So we have a few questions. 3 is a nice number. Let's start with one. Do you want to read it? Yeah.
HLA sequences are highly population specific. Have you seen the same for TCR, BCR? Is there an effort to ensure ethnic diversity in the population studied? Well, yes. I mean, this certainly is the case, and I'm almost tempted to ask Corey to answer this, because I mean, in the genomic work that I covered, which is the work out of Corey's lab in particular, there has been significant effort to make sure that lots of different populations are covered, and to use, for example, data from the telomere to telomere program, the Pan-Genome Project, which is ethnically labeled, and also to look for ancestry informative markers. All of those do provide good indications that there are significant differences. But I've got to your phone. Oh, there we are. So there is an effort. We do see it, and I'm sure there's a lot that we're missing as well. Are allelic differences being considered in the leader sequences and 5' UTRs? So the leader sequences are annotated on VDJ base, and it's an automatic annotation, but I think it's pretty good. It is automatic, though, so it's not always going to be perfect. There's a lot of data there. I would love somebody to pick it up and work very actively in this field. There's certainly very interesting variation there, and also correspondingly very little variation in the RSS, the other end of the genes, and that's really quite interesting too. UTRs, if anybody can help me annotate the UTRs in B genes, I'd really like to know about that. I mean, I've looked in general at tools for annotating UTRs, and they're not that good as far as I can see. So I'd like to do more there. I can certainly show you the sequence where I have it on VDJ base, but I can't convincingly annotate UTRs yet. Are allelic differences being considered? Are there any examples in which AIRR-seq and genomic-derived alleles differ? Well, we're putting together at the moment. We're putting together genomic and AIRR-seq inference data for around 180 rhesus macaques in IGH and light chains as well. And we are identifying differences. And sometimes those come down to just technical things. So we see, maybe we see a very, very low number of expressed sequences that cause a gene to be inferred, an allele to be inferred on the AIRR-seq side, perhaps isn't really real. So there have been some other technical things that have helped us refine. But we're actually beyond those things so far, I think, seeing very close correspondence between the two. It's something to keep a careful eye on. And perhaps one of the things that I meant to say when I put up that slide about genomic sequencing and assembly is it looks easy on the slide. It's not easy. It's taken a lot of work to get to the point that things are at. That's a lot of work on behalf of the team, and also a lot of work by the people that develop general purpose software for haplotyping and assembly. So don't assume that every genomic assembly out there is as good as the best sequences that you're seeing from Corey's lab today. There's a lot that isn't. And that's one of the places where it's very valuable to have the AIRR-seq data as well as the genomic data, because you can actually see things happen. You can see, you can see that several genes that are identified in the repertoire get collapsed to the same location in the genomic assembly because the genomic assembly just hasn't differentiated them correctly. Question on fire. You are using new V-segment names for the better germlines. How mad would you be if I mapped the names to the closest IMGT names? How bad is that practice? Well, I mean, not mad at all. I mean, the idea is, with existing sequences as well as new ones, to give them all, every sequence, a unique identifier to let you do that mapping. And in fact, with Rhesus, where we've been working on some internal germline sets, we deliberately publish a mapping between the 3 or is it 4 different naming systems for Rhesus, for Rhesus alleles. So not mad at all, but life's getting more difficult because IMGT are continuing to allocate names for neoalleles, regardless of what anybody else is doing. And so today, I think about a month ago, we saw that they reused a name for a human allele that we had given to an allele in our germline set. So I'm not mad at all, but you know, it's going to get difficult, I'm afraid. And I think that's it. That's it. Anything else? Oh, one more.
I'd be mad.
Rory has very high standards. What is the evolutionary reason for the allelic diversity in BCR TCRs? Well, I am not an evolutionary biologist, but if you want what's essentially a layman's kind of supposition, these genes are very similar. Breaks in DNA do occur. Repair mechanisms are not perfect, and I think that is at least one mechanism through which you can get not just the allelic diversity, but you can also get these big structural variants that we see, which can be massive in some cases. But what does it say about evolutionary pressure? I'd love to know. I'm much less sure about that, but I can't answer, I'm afraid.